Dear colleagues, dear friends, I would like to start expressing my sincere thanks to Maria Gloria Dominguez Bello and the Scientific Committee of the Global Microbiome Network, Peru 2021, for this opportunity I'm given to give this presentation. I will tell you about standards for metagenomics. I will essentially tell you of why metagenomics, why standards, and how, essentially which standards, going from pre-analytics to bioanalysis, and ongoing work on the comparison between shotgun and 16S data, and then just try to highlight possible futures in terms of clinical applications and moving towards the million microbiomes. So why metagenomics? We have unmet needs in diagnostic, in prevention, in therapeutics that could benefit from microbiome-based approaches. And expectations could encompass non-invasive diagnostic assessment of severity or risk, and there's many references for that. Prediction of disease onset that would require a large prospective longitudinal cohort studies, illustrated by the recent paper of the Rees laboratory in Belgium. Prediction of disease severity and aggravation risk. One example here in liver conditions. Prediction of response to treatment and companion diagnostic with illustrations from work being done in cancer immunotherapy. And rational preventive or risk mitigation measures in nutrition, prebiotics, probiotics, symbiotics, rationally design risk abrogation measures with live biotherapeutic products or fecal microbiota transfer in mind, and rationally design therapeutic measures and add-ons. Now, just to give an illustration, I'm emphasizing in this slide gene richness as a health stratifier. We started describing the distribution of humans as a function of gene richness within the MetaHit European program, and we were able to show that it will range from 200,000 up to close to 1 million genes in terms of gene count per microbiome. And you can see that the distribution is bimodal in populations like the obese or non-obese population. Essentially what we did observe since that time is that low gene count, which I call here posibiosis, is associated to more severe metabolic and inflammatory symptoms in overweight and obesity. It's associated with a non-response to low-calorie diet in overweight and obesity. It's associated with more severe or aggravating conditions in liver cirrhosis and non-response to immunotherapy in cancer. It represents 15% of the lean population, up to 40% of the overweight or moderately obese population, and among morbid obese awaiting bypass surgery, the low gene count population is 75% of the population. So in this particular case, it's really an indicator of the severity of. Now, in the absence of shared standards and references, we have the risk that different pipelines will tell different biological stories. And this is true even at the phylum level. I think we all know very well of this example illustrated by the comparison of American gut versus ubiome profiles for one single sample. And we have similar unpublished observations when we compare the human microbiome program overall characterization and the MetaHit European program characterization with a ratio of Bacteroidetes to Firmicutes that is essentially inverse. What we did within the European program IHMS for International Human Microbiome Standards is to mobilize 21 different laboratories worldwide for just the characterization of the best DNA extraction protocol. In fact, within IHMS, we did come up with standard operating procedures from all steps, ranging from sample identification, sample collection, depending on how much time the sample has to take before it reaches the laboratory for technical implementation, and DNA extraction. We also did provide soaps for bioinformatics, quality control, and so on. These are all on the Microbiome-Standards website since April 2015. For DNA extraction, we compared, as I was mentioning, 21 different laboratories, and we came up with only two soaps that were highly comparable, one of them amenable to automation. Expected profile. And when we work with different DNA extraction protocols, we have reasonably similar observations, and we do miss the eukaryotes in our experiments for the moment. Similarly with DNA coming from ATCC, where we are quite close to the expected results. Now, what we have initiated recently is what we call a ring test comparison between shotgun and 16S profiles. We're working on 5 samples from 5 healthy subjects, collected at 2 time points for each subject, separated by roughly 3 months. We call them S11, S12, S21, S22, etc. They all were derived from standardized sample collection and DNA preparation soaps, those provided by IHMS and performed by MetaGenoPolis. DNA extracts were shared among partners, and shotgun was performed by MetaGenoPolis, 20 million reads, proton reads, versus and compared to 16S V3, V4 region MiSeq Illumina sequencing 2 times 250 base pairs over 1 million reads, performed by 6 partners, P1 through P6. In fact, partner 2 was just reanalyzing the data, the 16S sequence data from partner 1. Shown here are the pipelines that were used, the clustering methods, the taxonomic database, and the essentials of the trimming. What we obtained from that is as expected when we look at shotgun data at the genus level by PICO-A based on Bray-Curtis comparisons or similarity. We have, as expected, each subject coming as showing as its own metagenomic profile with a rather high stability over the 3 months period. Subject 3, subject 5, subject 4, 1 and 2 are quite close to one another apparently in this comparison. And then when we plot everything together, what we see, I should say, unfortunately, is that the first differentiator is the laboratory where the data is analyzed. 2 of the 16S based profiles are very far from the rest, let's say, and interestingly, P2, which is the reanalysis of the sequence data from P1, gets much closer from the big crowd. But yet, in spite of that, this is work in progress, but the primary differentiator is the laboratory performing the analysis. So we are all telling somewhat different biological stories in the end. Similarly, a similarity between shotgun and 16S profiles is obviously highly impacted by the bioanalytical pipeline as well. And so our dream could be for the future that we move towards microbiome profiling and symbiosis monitoring in the clinics, i.e. that the medical doctor could prescribe fecal microbiome assessment at the same time as it commonly prescribes blood bioclinical measurements. And in this context, basically the expectation is that prescription of microbiome testing and integrated analysis would aim to detect intestinal dysbiosis or altered host microbes symbiosis, to monitor microbiome and symbiosis status over time, and to integrate microbiome and symbiosis status into nutritional and clinical management. Obviously, as we can see today, this will require standards, this will require more science, evidence base, and big numbers. And as we mention big numbers, we are at times that are quite interesting, with the inception in October 2019 of the Million Microbiome of Humans project. So essentially we're moving towards 1 million microbiomes being comparatively characterized, and in France, as part of the MMHP, we are initiating the pre-competitive 100,000 French microbiomes project, which we call the French GUT, to be simple, together with the Microbiome Foundation, as a citizen science approach. And we include cohorts and networks that are quite numerous actually in France at the moment. So to conclude, my take-home message is prescription of microbiome profile to assess the state of host microbes symbiosis in clinical practice will be the next frontier. The microbiome is a key driver of health and well-being of the human holobiont. Microbiome studies in the clinical setting are essential for the translation of corresponding innovations. Standardization is crucial to implement microbiome studies and to validate clinical usefulness of symbiosis profiling. And the microbiome will be sensitive and responsive to many confounders that have to be taken into consideration in clinical trials, and obviously it will have to be so in clinical applications. Let me thank you very much for your attention with these slides that just highlights many of the colleagues in the Michaelis Institute and the Metagenomics laboratory where I work at Jouy-en-Josas near Paris. Thank you again very much for your attention.